There we go. Okay, so we are recording. There's also an option now that you can have teams make a transcript of what you actually say. And sometime back in the fall, I was happening to record a lecture about male reproductive anatomy, and the transcript button was clicked and it recorded what I literally said, and it was hilarious on so many levels. I do not like to read the transcript of what I literally say because it's just very unsophisticated when you read it out in words compared to other professors, but also just the stuff that I was talking about was funny. And then it kept spelling, there was some word it kept spelling wrong all different ways. Like it might have been seminiferous or something like that, that doesn't know that word, it keeps putting different things. So today I do not click transcript, just record in general. So we know now kind of what's going on, that the dead smooth strain is leaking DNA out into the environment. And the living rough strain is going to use that DNA to build a capsule. And that's going to allow the rough strain to actually convert to a smooth strain and become this and kill the mouse. So DNA was the key factor that is allowing this to happen. It took many years and other people to help figure out that that's what's happening. And remember that this is not choosing what DNA it wants for this to happen. So any of these mechanisms for genetic transfer, it's kind of a crapshoot what DNA the bacterium will get. It might be good, it might not be good, but key fact is, and you can write this somewhere else on your page. If the new DNA confers a survival or a reproductive, or let's call it multiplying, since this is prokaryotes. If the new DNA is going to confer an advantage, that gene is going to persist and it will be passed down to the daughter cells. If it confers an advantage. So there could have been other rough strains mixed in that tube that got other pieces of DNA from other random places in this Streptococcus pneumoniae genome. And that gene didn't help them. And the mouse was able to see them and fight them off, and they were destroyed. But if the rough strain gets this segment of DNA, which is a very tiny piece out of the whole entire set of DNA, if you get that, that's the golden ticket. Feel like Willy Wonka? I do too. Golden ticket. I get to live. I get to hide from the immune system. So will all my offspring. We are the best. Yeah.
So what were the odds that happened the first time you did it to get that gene? Is that like crazy that it happened?
It's kind of surprising that it happened when he tried it. But he was using fairly thick cultures. But he might have tried it and it didn't work and then just move on to something else. Because it's kind of a chance. You wouldn't expect it to continually happen. But what made it more likely to happen is he had so many rough and so many smooth in those tubes that it made it more likely. Yeah. It's unlikely, but when it happens and it confers an advantage, then it becomes amazing for that organism that picked up this actually useful gene. So let's take a look sort of at what it takes actually literally to get this to happen. Oh, here's another example of how you can do this in a lab. So the way that this was kind of later proved in a lab setting is to take some bacteria that have a mutation in a gene for making tryptophan. So we're calling these trip minus. These are mutant bacteria that can't build tryptophan, which is an essential amino acid for people and for these bacteria. And if you try to grow them on agar that has no tryptophan, they won't grow because it's an essential nutrient. So that's the setup. If you take these same mutant cells that can't make tryptophan, they're mutants, and you give them purified DNA from cells that are normal or trip positive, they can make tryptophan. They got the genes to make tryptophan. And you mix that DNA with the cells and you just let them sit there. There's a chance. There's a chance. There's a chance that some of them will be transformed with the trip gene. Or trip genes, it's probably an awkward one. So you'll have some get the genes that they need. And you will have a few colonies show up on this plate that are recombinant. So they have gotten the golden ticket tryptophan genes, and you get a few. How many is this? Like 18? I don't know. It's 23. We're gonna say it's 23. 23 colonies. How many were in this tube? Hundreds of millions. So that's kind of the rate of how rare this is, that any individual cell will get the actual gene that will help it. And that's in a lab setting. So yeah, it's rare. But if you get the gene that helps you survive, you will survive. And that becomes evolutionarily advantageous. Evolutionarily advantageous. Said it correctly. Now let's look, what does it actually take? How does this work? The details of how it works, I am not so, this next slide is not one that you need to know a lot of details, but I want to kind of describe how it works and put some keywords up here. So we've got here a bacterial cell. This is its own chromosome in a circle. This is the DNA that came from the outside in purple. So for outside DNA to get inside the host cell, this is what's required. So what is required for transformation to happen with naked DNA from the environment? So this DNA is coming from the environment. It has to bind the new cell. And so they have on their surfaces DNA binding proteins, because the ability to do this is helpful overall. There will be one strand of the new DNA from the environment comes in. Just need one because of complementary base pairing. If you got one strand, the other information can be filled in. And then the new DNA must integrate and combine with the recipient cell. The recipient cell genome. If this DNA comes into the cytoplasm and it's sitting around in there single-stranded, that's just tragic. Like it'd be like having the ticket in your pocket that you don't know it's there. So if this DNA is going to get usable and heritable, it has to join the genome. And there is a particular protein that allows this called REC-A. REC-A enzyme is going to assist with the joining and integrating process so that the new DNA then is going to be stable. So then it wouldn't get destroyed if it's joined the recipient's genome. And then it is inheritable. It is stable, it is inheritable, and it is usable. That's what it takes. And REC-A protein is very important for that. So imagine that you are a ref strain and you pick up some DNA that is not a capsule gene, some other random gene. And by miraculous chance, you take it in, REC-A does its work, the new gene gets integrated in the genome. And that's already rare, but it's likely that whatever that is, it's a gene you've already got. Something that's not going to help you. You got it now, but it's kind of pointless. Wouldn't help you. This system is chance built upon chance, built upon chance. So it's a multiplying of rare probabilities that this can even work. But when it does work, if it confers survival advantage, that's a big if, this cell will survive something that normally would kill it. That's when we see the effects. And over long periods of time, this is a major way that bacteria get new genes and are, assisted to evolve, I guess you could say. But it takes a lot to get that. It's chance built upon chance, built upon chance. REC-A is important for this process. REC-A is important. The details of what it's doing, just don't later. I don't like to have a slide without a picture of it. Maybe I should have. So if this new DNA is not integrated, it will get destroyed in the cytoplasm. Single-stranded DNA that hangs around in the cytoplasm is fairly pointless and will get degraded quickly. So just because you get into the cell, you still have to get joined with the genome to have a chance to be useful or have a chance to get passed down. Molecular details of that, we don't need to know for this class.
How does only one of the two strands get in?
I don't know.
Or how did-
I don't know if there is a selection process of which strand.
So it just breaks the helix. and just-
Yeah, so let's go back to previous picture.
That protein's.
The book you just sent that. It was good.
So these. There is a binding protein and likely it is in a complex with a nuclease. Some of this is just. It's easier to get one strand in than two. And if you've got one strand, the rest can be filled in by base pairing. So to get it to one strand, there's a nuclease that cuts up the other strand. I don't know if there is a selection, like which strand. That probably is known by now, but I don't know it. And so these would be in a complex together, getting the DNA to come in. And then once it is in, there are some stabilizing proteins that can kind of keep this from being destroyed right away until Rec A can work on it.
Okay.
This is a big, important research area in bacterial genetics. Like, how does this work? People have figured out how to use this to make bacteria do what we want by giving them new genes that we want them to have. This is actually super easy to do in a lab. I think you're doing, no, none of you are molecular. If you take molecular bio, you will do this in a lab setting. So before we move to the next type of DNA recombination, I want to add some info about what types of DNA can move during transformation, because this is going to be different for the different methods. So for transformation, the DNA can come from any source. The DNA can come from any source. It doesn't have to be bacterial. It doesn't have to be in a circle. It doesn't have to be any particular length. The DNA can be a fairly long piece. It is known what is the maximum length, but I don't know that myself. It can be a fairly long piece. And if this DNA does not have introns, if the DNA does not have intron sequences, the bacterial cell is generally going to be able to use it. So if we want to use bacteria to make eukaryotic gene products, you have to take out the introns because bacteria can't deal with that to produce the product that you're trying to get them to make.
So it can't splice the exons out.
It can't splice exons. Yeah. Yeah, I think that's it. Yeah, that'll work. Okay, so that's transformation. Normally I know I do this in nursing micro. I think on your all's test, there will be a decent sized question about transformation in Griffith's experiment as one of the big questions. These other ones, they are going to be on the test in multiple choice, but not in as much detail. But you do want to know what makes them different, what characterizes them. Okay, so the second way that genes can move between bacteria is by transduction. And transduction is a viral method of moving DNA in between cells. And we've already kind of seen how this happens, actually. But the DNA that leaves one cell and travels to another by a virus method, that's called transduction. And during transduction, what is happening is a virus picks up bacterial DNA by mistake. So at the end of last Thursday, we talked about that if you're a bacteriovirus, bacteriophage, you pack your head full of some DNA and then you leave. You don't check what it is. It's like, do I have enough DNA? Okay, yes. Off I go. I got some DNA. Ready. And once in a while, this one, we want to circle this one. That virus right there has a different color of DNA inside it than the second from the left.
There's,
So as viruses are in the cytoplasm doing assembly, there is a chance that one new virus will get made that accidentally has bacterial DNA in its head and not viral DNA. And so this one, now, if it goes and infects a new cell, it is bringing it bacterial DNA, not viral DNA. Let's see if I've got another follow-up picture. Yes, okay. So before I erase this, a normal virus comes and infects this cell, puts in its genome. As the host cell is dying, there are pieces of host cell genome that are floating around in there. One of them accidentally gets into a new virus. This virus now is wrongly constructed, but it could go and pass this DNA to a new host. That's the situation. So the virus is using normal lytic cycle. So now we're going to follow special green virus. What happens next? So here it comes. And if you are this cell, this amuses me to think about. Like if they can't tell that the virus has come to them, but not only are you not going to die, you are getting something new. You're getting some information. So great, you're not going to die, but even though a virus has just infected you, and this may or may not be useful, but it's something new. It could be a job application to legacy theater that you don't care about what you do. I think that would be an okay job. Weekends. Yeah. Or it could be a paper about your child is going to do Shark Tank at school, big project. Or it could be blank checks. You don't know, something new. It still does have to integrate. So if this DNA is going to be usable, it does have to integrate with the new genome. And then once that happens, we call the cell that just got this DNA, it is now transduced. This is transduction. So the way that I remember transduction is like the virus has to duck down, transmit that DNA. That's stupid. That's how I remember transduction, as apart from transformation. So transduction has a lot more requirements on it. So for transduction to happen between two cells, the donor and the recipient have to be, the donor and the recipient must be infectable by the same virus. And if you are infectable by the same virus, that means that the donor and the recipient cell have to be fairly closely related. So, for example, this would be a way to move genes between types of E. coli. This would not move genes between E. coli and Staphylococcus aureus because they're too distantly related. They don't share viruses that infect both of them. The length of the DNA is constrained. The length of the DNA must fit in a virus. And the DNA has to be linear. So transduction overall is kind of less common in nature than transformation because it's got more requirements. Still, the DNA that is moving is not chosen by anyone. It is a random crapshoot of what DNA is moving.
Do we know the difference between lytic and lysogenic transduction?
Lysogenic transduction would indicate that this host DNA, you don't need to know that for this class. Just transduction generally. It's a virus. The cells have to be co-infectable by the same virus.
So for transduction, it goes through the lytic cycle like normal, and then it does its transduction.
Yeah, the lytic cycle is required for this DNA to ever get out of this thing. So it could have been in a lysogenic cycle at some point, but it can't leave until there is a lytic cycle where this can leave. Is that answering what you're?
Yeah, I was making sure the lytic cycle like had to happen first for the transduction to,
Yeah,
you know.
yeah. I think I actually want to save conjugation for Thursday and use this time right now to talk about exam topics. Conjugation is on the exam. It is not a major exam topic. So I'm going to take a few minutes and collect my thoughts, make sure I don't leave out any major topic that's on this next exam. this a little bit. Okay, so exam 2 is probably the hardest test. Exam 3, it looks like this is a lot of like boring stuff. I don't ask as difficult of questions in exam 3 because I know that people are not naturally excited about this. And it will be interesting the way that I teach it. Exam 2 is kind of the hardest test, and it is most like a cell test. So I was pretty happy overall with y'all's exam 1. Exam 1 was not a huge amount of thinking. It was a lot more memory work. This test has less memory and more application. So just something to know. Some of you have asked me in different formats or put on the survey, like that I word things vaguely, and I know that I do. So during this test or during reviews or whatever, don't hesitate to ask me to completely rephrase the question. Like actually in all my classes, I'm happy to do that. Completely happy to do that. So the things that's kind of the big questions that are on this exam is you will have for the board picture is going to be lac operon. And that's where we have that cartoon that's got the lac genes and the repressor and the operator. That's going to be up on the screen. And you'll have that available to look at. So there will be several questions on lac operon that are like the practice questions. And I'm sure on Thursday that some of our time will be spent going over some of those. So that style of question. That will be probably about 20% of the test is that stuff. It is multiple questions. There is partial credit. So you all have done well at not leaving stuff blank. I want to like throttle AMP2 students. Do not leave this blank. Do not leave this blank. Do not leave this whole page blank, even if you weren't there that day. So that's a big one. So that one is probably about 20 points. The stuff out of this day relates to this. Like it is related to this, and it is related to all gene things that we've talked about, but there's not a particular big question about that. There's maybe multiple choice or something. So viral structure and replication, there are some memorizing questions there. So you need to know what are the viral parts / components. Like they all have to have a genome. They all have to have a capsid. Some of them have a spike. Some of them have envelope. There are 5 of those. You also need to have memorized what are the life cycle stages. There are 5 of those. And normally I pick out one of those and ask you to list them for 5 points. And knowing that stuff is going to help you on the rest of the test anyway. The other big topic out of that section is what is the difference between lysogenic versus lytic. That is not a huge amount of points on the test, but it's just really important that you study that some because it relates to transduction. It relates to a lot of different things. That's from that day. From bacteriophage T4. generally that I ask some kind of question about how does an early protein or a middle protein or a late protein. I don't ask about all of them. I usually just pick one. How does it get made early? Or if it's a middle, how does it get made in the middle? What's required for that? And as part of this question, you need to know some examples. Like what is one of the early proteins? What is one of the late proteins? So that's a fairly medium-sized question. You won't have the picture on that. That one will need a little bit of study. Influenza, the major topic out of influenza is drift versus shift. And there will be other questions about coronavirus and influenza, but the big concept is what is the difference between drift and shift? How do they happen? And what type of things are going to result from it? That one, I have a little bit to say on Thursday, of like relating that back to coronavirus. Like when did we see shift in coronavirus versus when did we see drift? And then on gene transfer, there will be kind of something about compare, contrast, transformation, transduction, conjugation. And then a detailed question about Griffith's experiment. This test might have less multiple choice than the last test. I think test 2 has less multiple choice, test 3 has more multiple choice. Test 4 is a take home and completely different.
When is our lab? Sure.
Lab exam is the Wednesday after spring break. This one's next week. Then there's spring break, then the lab exam.
Will we be doing like another lab exam after?
No, there's only one lab exam.
Are we still doing lab after?
There still will be lab, so about half of that time is you working on your unknowns project on your own pace and stuff, and that's about 2 weeks. And then so at the end of the semester, we have one actual lab, but we use that time for kind of like group learning. And there's something called flipped classroom. So flipped classroom is when I have you all read and watch videos of certain stuff, and then when you come to class, it's not me lecturing, it's you working on stuff. And so if you can get past, well, like basically the end of March, April in this class is amazing. So if you get past the end of March, April is amazing, super fun, more relaxed. And this may look awful, but I teach this in anyway. Yeah. What week is this? Is this week 7?
Yes.
I think midterm grades are due next week, so. Yes.
Oh, we're not going to have a pilot test grade. No shot. There's no way we're gonna have that test grade.
Before midterm?
No way. Yeah, I don't know, maybe. She's telling me prep for lag.
I did not see it.